In medicine today, we are just swimming in genetic data. It's an absolute mountain of it. But how do you actually turn all that raw information into a clear story? How do you find the villain, the specific biological pathway that's actually driving a disease? Well, that is the code we are cracking today.
Okay, so imagine you're a detective. You've got two different crime scenes, in this case, two different types of cancer. And you have thousands, literally thousands, of potential persons of interest, all these different genes. The raw data is just this massive, overwhelming list of names. So where do you even start? How do you find the real culprits and figure out what makes these two cancers tick?
Every great detective story needs a case file, right? And ours comes from a landmark study in cancer research. We're diving into a really famous dataset from a study by a researcher named Golub and his colleagues. Now, they had genetic data from patients with two different kinds of acute leukemia. One is called AL, the other is AML. They knew these diseases were different, of course, but the real mystery was buried deep inside the genetic code. Their whole goal was to find the exact genetic patterns that made each one unique.
And when I say the mystery was buried deep, I mean it. Scientists weren't just dealing with, a handful of genes. For every single patient, their list of potential clues, what they call their gene universe, started at over 5,000. That's 5,421 potential leads to follow. It's a huge number.
So, to solve a case this complex, you don't just need a magnifying glass. No, you need a totally new way of thinking. You need a pattern detective.
And that detective has a name. It's a method called Gene Set Enrichment Analysis. The real genius of this approach is that it doesn't get lost in the weeds, looking at every single gene one by one. Instead, it looks for conspiracies. It hunts for groups of genes, all part of the same biological team that are all acting suspiciously together.
So how does this detective actually work? It's a pretty brilliant four-step process. First, you start with that huge pool of persons of interest. That's all the genes. Next, you run a test that narrows it down to a shorter list of key suspects. These are the genes whose activity is way different between the two cancers. But here's the crucial step. You check their known affiliations. You ask, what do these genes actually do? And that's when you find it. A suspiciously large number of your suspects all belong to the same few biological gangs. That discovery, that's what scientists call enrichment.
Now, a detective is only as good as their sources, right? To check a gene's so-called affiliations, scientists need these huge encyclopedias of biological knowledge. And they mainly use two big ones. The first is called Gene Ontology, or GO for short. Think of this like a gene's job description. It tells you the specific task it performs in the cell. The second is the KEGG database. This is more like a team roster. It shows you which larger biological pathway, or team, a gene plays for. Using both of them together gives you a really complete picture of what each of your suspects is up to.
Okay, so we have our case file, we have our detective method, and we have our encyclopedias. It is time to put it all together and really start connecting the clues in our leukemia mystery.
The very first step is to find our primary suspects. The analysis sifts through all 5,421 of those genes, and it flags only the ones that are upregulated, which is just a fancy way of saying they are working overtime in one type of cancer compared to the other. These are our hot leads.
And the results start rolling in. For AL leukemia, the analysis flags 329 distinct genes that are working way too hard. This is our first list of suspects.
And for AML leukemia, it finds a totally separate list of 268 overactive genes. So now we have two different lists of suspects. But here's the thing, a list of names isn't a story. We still need to find the pattern.
And this is where the magic happens. This is the part where we go from a boring spreadsheet full of gene names to something you can actually see and understand. We're about to create a map of the entire crime scene.
This really is the aha moment. All that incredibly complex data analysis, all that work, it pays off in one single powerful picture. We're about to see the hidden biological machinery of each cancer just laid out right in front of us.
Okay, before we see the final map, here's the key. You remember those lists of suspect genes? The analysis checks what gangs or biological processes they belong to. On the map we're about to see, each dot, or node, represents one of those processes. Red dots are the processes being run by the ALL-specific genes, and the green dots are the processes run by the AML-specific genes. Simple as that.
And here is the map. I mean, wow, this is no longer just a list of genes. We can actually see two completely distinct criminal operations at work. Look at that red cluster. That's the whole interconnected network of processes that's driving all AL leukemia. And now look at the green cluster. It's a totally separate and distinct network that's powering AML. It is a stunningly clear picture. Two cancers running on two fundamentally different biological engines.
I mean, this is incredibly powerful stuff. But what's the ultimate point of drawing a map like this? Well, it's about turning all of these clues into a path forward.
Here's the crucial takeaway. This kind of analysis completely transforms our understanding. We move from just a flat list of genes to seeing the disease's unique personality, the very systems it relies on to survive and grow. And by understanding that core machinery, scientists can ask much smarter questions. Instead of just guessing, they can look at this map and say, hey, what if we target this specific node right here? Can we find a drug that disrupts this one process and makes the entire enemy network just collapse?
And that is the ultimate payoff of cracking the genetic code. It doesn't just solve a mystery about what a disease is. It gives us a treasure map. It points directly to the biological hotspots, the pathways that could one day become the targets for brand new, life-saving therapies. The investigation, as they say, continues.